Okie dokie. So I think this is where we left off. Um, yeah, so we've been sort of going through exploring broad topics in how chemicals can get into different components of the environment, likely issues that they may cause. And we've been talking about water. And so we wanted to talk a little bit. We just finished talking about the difference between point source solution and non-point source solution, or pollution, not pollution, pollution. And so one of the particular issues with pollution into water bodies, whether it's point source or non-point source, is that um, the introduction of especially organic pollution, and when we say organic here, we mean more pollution in the biomass organic sense rather than the organochemical sort of sense, but the organochemical sort of sense of pollution can also cause similar issues. Um, but one of the consequences of that introduction of organic pollution is it can affect the biological oxygen demand, there we go, of the aquatic environment. So biological oxygen demand, you can think of this as how much oxygen is needed to be dissolved in the water in order for everyone in the water to have enough oxygen to respire, to breathe, to do respiration, to extract energy from compounds. Some organisms require more oxygen than others. This can largely be a function of size. Larger organisms can require more oxygen than smaller organisms. You have a fish versus a bacteria. The fish clearly needs more oxygen than the little bacteria does. But even within fish or within invertebrates that might be in the water, or even among bacteria, different species can require different amounts of oxygen. And in an aquatic environment, you typically have a variety of organisms that have a variety of oxygen demand. Some require more, some require less, at all sizes and ranges. And when you introduce organic pollution, you can muck with that balance of available oxygen. So on this diagram here, we've got an aquatic habitat with all of the little individuals that exist in this aquatic habitat. And we first observed these organisms at this, either we can think of this as time point zero or distance point zero, where we've got sort of normal, general status quo habitat, no organic pollution yet introduced. And so what we see with those organisms is that there will typically be that diversity, that variety of organisms that have a variety of oxygen demands. And typically, when there is no organic pollution introduced, the available dissolved oxygen is pretty high, suggesting that everyone in this environment has enough oxygen to do what they need to do. And because the available oxygen is high, the biological oxygen demand, this white line, is going to be low. Everyone's just sort of doing their normal thing. There's nothing extra going on. And so we don't need extra oxygen for things. You can kind of think of these black and white lines as the black line is your bank account, like how much money you have in the bank, versus the white line, which is how much your expenses are. So when everything is going normally, you've got plenty of money in the bank, hypothetically, and your bill load is comparatively low, hypothetically. And then something happens. We have some sort of pollution event. If this were a river, this might be at some point later downstream on the river from this point. If this is a lake, we might think this is some time t later from where this base condition was. And so this organic pollution, whether it's sewage or whether it's maybe an organic petrochemical, is introduced. And when that organic substance is introduced, you can see that our lines reverse. And so now with all of this extra organic material, what is happening with the introduction of this extra organic material is we're really ramping up respiration, especially for bacteria and really small invertebrate type organisms, because there's all of this sort of extra schmutz in the water that needs to be broken down, consumed, and gotten rid of. And so what we see at this point when we introduce some organic pollution is we see those black and white lines switch. So now our bill expenditure is high, right? We've ramped up respiration, and so we need more oxygen to do the more respiration that we've now induced with this organic pollution. So our biological oxygen demand, this white line, is now high. And because we're now spending more money, spending more oxygen to do that extra respiration, the amount of available oxygen is low. The level of money in our bank account is dropping because we're spending it all on this bill of breaking down this organic material. As we do so, as we flip those lines, spending more oxygen than we have oxygen available, we can see that the composition of organisms is going to shift to accommodate the amount of available oxygen. And so we will see a shift away from a wide diversity of organisms to a highly restricted set of organisms. And these are going to be organisms that are capable of living in an aqueous environment with very low available oxygen. So they can deal with very, very low fun. They're cool living on ramen. They're not unhappy about that. They can make do. Everybody else who needs something a little bit more nutritious than ramen has gone away. They've left this area where oxygen has been depleted. And what we can see then, either through time or distance, so we can either think of this as time after the initial pollution event or distance away from the pollution event, is that the farther we get away from that pollution event, we gradually see these lines sort of come back to their original point in time, where we have high concentrations of oxygen available for everybody to do all the respiration that they need to do, and we have very low extra demand on that oxygen. So increasing pollution can have these really drastic ecosystem shifts as we muck with the amount of available oxygen. This kind of flip with these black and white lines here, like we see in this polluted zone, this is the kind of situation that we often see in aquatic habitats where an oil spill has occurred. So we saw it in the Gulf of Mexico with the explosion of the Deepwater Horizon. We see it in other water bodies when oil pipelines spill and explode. It's basically, just a giant dumping of organic material, i.e. oil, into the aquatic habitat. And there are microorganisms that can break down that oil, but they need oxygen to do so. And so we see this difference in white and black lines in those polluted zones become extremely exaggerated. And I think I've got a diagram later on that shows a little bit more clearly what that can do in ocean habitat. All right, so that I think finishes up a little bit of our organic pollution that we were talking about with point versus non-point. To bring it back to some more inorganic stuff, inorganic pollutants can also affect water. Lots of different kinds of inorganic things. One of the most common forms of inorganic pollution that can affect water bodies is the introduction of acids or bases. Acids is more common than bases. You don't often hear about a spill of basic material, that causes an increase in the pH of the water body. Usually it's some sort of acid. Acids are especially introduced as a byproduct of mining activities. Sorry, I just want to try and skip ahead because, oh yeah, all right. I just don't want to say too much about the little vignette that I have planned for that. All right, so we'll just leave that there for now, as that acids are introduced as a byproduct of mining. We can also have acid introduced through acid rain deposition, right? So when acid compounds like those NOx and SOx, right? So when you think acid rain, you want to think about those nitrogen oxide kind of compounds with some unknown number of oxygens. That's why we call it NOx. SOx, again, sulfur with some unknown number of oxygens, variable number of oxygens. That acid rain that can be generated from the combustion of fossil fuels that then goes up into the atmosphere. Those acid compounds, the NOx and SOx, attach themselves to rain droplets. Those rain droplets fall onto the landscape. That landscape can be terrestrial, just dry land, or it can be a lake or a river. And if it's a lake or a river, then it's these NOx and SOx compounds going into the lake or the river, thereby lowering the pH. We've seen some consequences of acid rain deposition in lakes in the Adirondacks. It's been a topic of concern for at least the last 20 years. In the Adirondacks, not only in the Adirondacks, but also New Hampshire. I think it was, I always forget the name of the lake right as I'm about to say it. I want to say it's Lake Winnipesaukee in New Hampshire. Used to be one of those lakes that was considered dead because it had been so acidified by acid rain deposition from coal-fired power plants releasing NOx and SOx into the atmosphere that would then rain down on the lake. The lake had actually reached what we call clear water status, where it had become so acidic that nothing could live in the lake, and so the lake was just like pure clear water that you could see all the way down. There was no microorganisms, there were no fish, there were no plants. And through lots of rehabilitation work, Lake Winnipesaukee has now come back. It's a totally fine lake. You can fish in it, you can swim in it, you can do all the things in it. And that's thanks a lot in part to changes in the filtering process of emissions from those coal-fired power plants, changes over from coal to things like natural gas, improvements in filters, improvements to the surrounding terrestrial landscape to better filter out that acid as it runs into the lake. So we can rehabilitate lakes from acid rain deposition and rivers from acid rain deposition. So it's not a, well, that's it, it's trash, forget about that lake. No, we can fix these things. In addition to acids and base, one other large group of compounds that can affect water bodies include salts. Excess salts in environment can cause a lot of problems. And this is part of the work that I'm doing right now with my research team, is we are investigating the consequences of winter road salt utilization on stream organisms chronically over the course of a year. So we always think about road salt as it's a wintertime issue. We spread the salt in the winter, and when it's not winter anymore, we're not spreading salt, and ta-da, it magically goes away. We don't think about it anymore. But how many times has it gotten to be like April or May, and you walk by on the sidewalk and there's just like a pile of salt just hanging out on the sidewalk? That salt still has to get in. Or still, it doesn't just vanish in June. Nobody actually sweeps that stuff up. It just eventually rains so much that it dissolves and it gets washed into river bodies. Similarly, all along roadsides, that's happening. And so all throughout the non-winter season, we have road salt slowly leaching in and depositing in our water bodies, even though we're not actively spreading road salt. Some potential consequences of excess salts in our freshwater systems, especially, is, we can see excess sodium in people. There was a recent study out of Canada that excess sodium in, was it well water? It might have been well water. Excess sodium in well water from road salt had reached high enough concentrations that people with heart disease were being affected by the excess sodium. So excess sodium can cause health problems in people. It does cause a lot of problems in wildlife. We know that it can alter development of juvenile organisms. We know that it can interfere with cell reception signals going on, especially between microorganisms and other small organisms that use sensory chemical compounds to communicate with one another. It can reduce life expectancy, reduce reproduction. It can cause a whole lot of harm in that way. And that's from excess road salt. Road salt, we usually think of road salt as being just sodium chloride, but there are other forms of road salt that we typically use, like calcium chloride or magnesium chloride. They also cause similar problems, namely because of that chloride component of those salts. So some of the stuff that I'm doing with our research project is we are collecting bugs from streams and algae from streams all throughout the year, and we are going to not only look at their biodiversity, because no one's actually looked at the biodiversity of things before. We usually sort of just take one or two organisms and expose them to a whole bunch of salt and just kind of see what happens. But we haven't looked sort of ambiently at how many different kinds of bugs are in these streams with different concentrations of salt, or algae with different concentrations of salt. And we know that it can affect reproduction and development and whatnot, but we don't know a whole lot about why it's affecting development or why it's affecting reproduction. And so some of the other stuff that we're going to do is look at the biochemistry of these guys. How much carbon do they have? How much nitrogen? Nitrogen usually indicates how much protein content. And of course, my forte to look at the lipids, because lipids are really important in cell membrane integrity, especially neural cell development, which is especially important for fish, vertebrates, and see how we're affecting those lipid profiles with road salt. So road salt can be a really big issue. Increased salinization doesn't only come from road salt, it can also come from overuse of agricultural lands. So as agricultural fields are sort of used consistently, the application of fertilizers and the mixing of water, because you got to water your crops, you got to fertilize your crops, that chemical reaction in the soil can leave residual salts behind. And so if you've ever worked in a garden and never sort of taken the time to like let your garden rest, you'll notice that you can get this kind of salty crust on your garden soil, and that is residual salts left over from water and fertilizer combined. And that's where things like magnesium sulfate come into play, and ammonium chloride can come into play as those agricultural fertilizer residual salts. And of course, last but not least, related to the acid from mining, we can get actual metal contamination, mercury, lead, cadmium, the usual metals. All right. Sorry, lots of story time. Okay, so specifically to go back to the acid deposition, there was, right before I started here at Daemen, there was this catastrophe issue that happened in Colorado on the Animas River. So in 2015, on the Animas River in Colorado, and so here are some pictures of the Animas River. This is the Animas River before this happened, and then here's the same spot after this happened. In Colorado, there is a place called Silverton, and in Silverton was this place called the Gold King Mine. The Gold King Mine had opened in 1887 and was decommissioned in 1923. So it hadn't been used. So we're almost 100 years after the closure of this mine. This mine hadn't been used in 100 years. No mining activity, no leaching or no industrial activity, no explosions of anything, just 100 years of nothing going on, except for all the stuff that was going on in the Gold King Mine. So when you have a mine, if you've ever watched like an old Western, you know, a mine is pretty much exactly what you think it is. It's a chunk of mountain that we've burrowed into and created all of these caverns to get at the minerals buried in the mountain. And when we stop mining that mountain, those caverns stay. You know, maybe you board up the entrance and you put danger on it, don't go in, and, you know, some kids looking for their lost dog get lost in there, you know, whatever the Western movie it is that you've seen about a mine. But other than that, boarding up, those caverns stay open, meaning all of the minerals and compounds in that rock are also exposed to the air. And not only are they exposed to the air, but there is groundwater leaking through the mountain. So we always think of groundwater as just a sort of down low thing, but groundwater follows the ground, and mountain is ground. It's just ground with contour. And so just to have an excuse to draw badly, so let's say we've got a mountain here. So we've got a mountain and we've burrowed in and we've made a mine. So here's a mine. The groundwater is still going to be there doing its thing, following the mountain. And so when you have these mine caverns that you've opened up and left exposed, that groundwater then percolates into those empty caverns that we have created. And that groundwater is going to interact with the minerals in the exposed rock and the air in the exposed rock. And when you get water and air and minerals all interacting together, you get chemical reaction. They take a long time to happen. It's not as fast as a chemistry lab, but chemical reactions do happen. One of the chemical reactions that can happen is acid creation. So this mine closed in 1923, long before we really sort of highly mechanized the use of acid in metal purification for mines. But acid was nonetheless created. It was created as groundwater interacted with pyrite left in the rock mineral with the water coming through and the air, and this created this acidic drainage. So pyrite, otherwise known as fool's gold. If you've ever, gone searching for fool's gold, it can be kind of a fun activity. I know growing up as a kid in the desert, we had lots of fool's gold as just part of the sand. And in the desert, we had sand everywhere. And it was a very common grade school activity to just comb through the sand looking for chunks of pyrite because we were bored children. But pyrite is made out of iron sulfide. That's one of the main components of pyrite is iron sulfide. And so when you mix water and iron sulfide and air, you get iron sulfuric acid, which is this nice sort of milky yellowy color. And so this mine was filling up with this milky acidic iron sulfide acid drainage coming out of the mountain. And the EPA knew about this. So the EPA knew about this. In 1990, they took over management of the Animas River site and the Gold King mine. They took over control of that from Colorado. They tried draining it, so they had dammed up the rest of the mine to keep the acid in place, and they had set up drainage to kind of, slowly and controlled get this acid leakage out of this mine. And then in 2015, there was a landslide, and that landslide buried those pipes. And so you can right here kind of see the remnants of one of those pipes coming out of the mine. And so there was a landslide in 2015, and they're like, well, we've got to uncover these pipes so that we can get to them and check on them. And in the process of using bulldozers to remove the landslide so they could check on the pipes, they ruptured the pipes. And so, this ruptured the pipes, and it leached. It just spilled all of this iron sulfide acid right into the beautiful Animas River. It was a whole, to be blunt, it was a shit show. The director of the EPA at the time, Janet Napolitano, really botched, especially the communication with the public about this. They were not forthcoming with all of this. They weren't like they just sort of left it as, we don't know what happened. It's like, what do you mean you don't know what happened? This is your site. And then, all of this, you broke this, like when construction workers rupture a natural gas line, and you're like, come on, dummy. But it's kind of the same thing. And so they tried to brush it off. They was like, it's not a big deal. And people were looking at the river going, no, this is a big deal. And everyone was very worried about it. And they're like, yeah, it's fine. Don't worry about it. So it was an issue. But it's since been all cleaned up. The Animas River is once again nice and clear and blue. You can go kayaking and river rafting and fishing, all of those things in it, and it's all fine. They've redone all the drainage for the Gold King mine so that hopefully this won't happen again. Extra security measures in place so that if another landslide happens, they won't break pipes again. So it's hopefully all fixed up. But this is the kind of issue that can happen, especially with acid contamination. All right. So how do some of these things get into the water other than major disasters like this? Pollutants can get into waterways other than disasters through improper disposal of industrial and household wastes. So, especially if you've got older folks who are still kind of doing things the way we did them in 1960, a lot of disposal practices consisted of pour it down the drain with lots of water. So pour it down the drain with lots of water, or if not, down your sink drain so that it goes to a water treatment plant. Some people would pour it into a gutter and maybe just run the garden hose a little bit. And a lot of our sewers just go right back out to water bodies because they're meant for rainfall and not for treating water. So improper disposal of things, and if not down a drain, old advice on how to deal with toxic substances in the home was to go out into your backyard, dig a hole, pour whatever into the hole, and then fill up the hole with dirt that you dug out of it. And you can imagine that if you did that and then it rained, stuff could leach out of the hole that you dug, you just poured your motor oil into. So ways like that are ways that things can get into water bodies. We can get runoff from all manner of things. Golf courses are especially notorious for leaching pesticides into water bodies because they want that perfect monoculture of whatever grass it is that's on a golf course. And they will spray and treat to kill anything that is not that perfect monoculture of grass. And of course, you don't want to be bitten up by mosquitoes or ticks while you're out there golfing. So they'll spray insecticides all over the lawn to make sure that you aren't eaten up by insects while you are golfing. So there's lots of chemicals that are used in golf courses. Other farms, very similarly. So here's a nice farm that's right along a river. So you can imagine if an insecticide is being tilled directly into the soil, that can leach right into that river. Once these crops start to grow, if they decide to dust with maybe some fertilizer or some other insecticide or an herbicide to kill some unwanted plants, that can again go right into the river. A number of years ago, the EPA estimates that about 500,000 metric tons of pesticides are used annually in the US. I'm sure that number is slightly different now. Hopefully it's gone down. I haven't double checked on it in a while. And on average, we've got about, let's assume we've done some work since I made this slide, and we'll call it 20,000 waste sites. So the EPA is aware of at least 20,000 sites that have toxic waste accumulated that are especially adjacent to water bodies that need to be dealt with. And there are a number of studies that have shown that even small amounts of some of these compounds can cause birth defects, death, and other serious health issues. So toxic substances can be a big problem in water bodies. We can also have degradation of water bodies that is not a chemical compound, particularly sediment and alterated temperatures. So sediment can be a big problem in water quality. Sediment, we often think of it as natural. It's a natural part, it's a normal part of river and lake function and processes to have sediment in them. It can bring nutrients from terrestrial landscapes into those aquatic water bodies. But it can cause internal shading that can decrease the photosynthesis of plants. It can scrub algae off of rocks, which reduces food sources available for fish and invertebrates. It can reduce the homes of invertebrates. If you have a nice cobbly stream, there are lots of invertebrates that really like living in between all of those little cobbles. This is also why if you are hiking along a river, you should not build a little tower of rocks in the middle of the river. As cute as it looks, it does destroy invertebrate homes. So please don't do that. But if you have a nice cobbly river and then it gets flooded with sediment, that can fill in all of those little crevices and destroy the homes of all those little invertebrates, little fish. You can also clog industrial equipment, especially like water intake valves for municipal water supplies. Heat is a less common water quality issue. It does happen, but we are increasingly seeing it less and less, thankfully. Usually where we get thermal pollution is the effluent from industrial cooling structures. So lots of industrial plants of various types are built on rivers so that they can use the flow of the river to come in, cool their equipment, and then the spent water then leaves downstream, carrying the excess heat from that equipment with it. That increase in temperature can kill aquatic life, especially very sensitive fish. If fish are accustomed and adapted to a specific range of temperatures, and then you really jack up the temperature, then you can kill those organisms. One of the places where we really see this issue of thermal pollution is especially associated with nuclear power plants. So nuclear power plants use water to keep their nuclear reactors cool. This is why we often see nuclear power plants built near water bodies. So you can go to Oswego, and there's a nuclear power plant in Oswego right there on Lake Ontario. 3 Mile Island down on the Susquehanna River in Pennsylvania, which they just turned back on, fun times. And this down here in the lower Hudson, this is Indian Point. So this is what the Indian Point nuclear power plant looks like. So this is just a satellite image that I took from Google Maps. This is the outstream from that cooling. So water would come in upstream up here, and then that water flows around the outside of the nuclear reaction chamber. So this river water is not interacting with nuclear radioactive material. I just want to make that clear. This water is not interacting with radioactive nuclear material. It is getting, however, very, very hot, right? Nuclear reactions generate a lot of energy. It makes things very, very hot. You've seen the aftermath of Chernobyl. You saw huge metal structures just liquefied. It's a lot of heat, and water can take all that heat. But now this outflow here then becomes extremely hot itself. And so you're taking this really hot water and mixing it in with this cooler river water beside it. And that, warmer than average temperature can cause heat shock, almost like a fever. A lot of organisms have heat shock proteins that they can deploy to kind of insulate themselves from that thermal shock. But it takes time to generate those heat shock proteins, and you can only make so much of them, and they can only absorb so much heat shock. And if the water temperature coming out here is too high, then those heat shock proteins aren't going to do you any good. Let's see, other current issues in water pollution also come to us from Jackson, Mississippi. So, excuse me, in Jackson, Mississippi, this is their municipal water, so not exactly a water body, but this is the water that they supply to the city of Jackson, Mississippi. So Jackson, if you're not familiar with Jackson, this is a city of about 150,000 people, not a terribly large city. But the demographics are that it is 80% people of color living in Jackson, Mississippi. There was a dramatic shift in those demographics in the 1980s. This caused a reduction of the tax base, and that left very reduced funds available for the maintenance of city services, and city managers who are predominantly white had to pick and choose which city services they were going to maintain and which ones they weren't. One of the ones that they decided to not maintain was the city's water infrastructure. And so some of the problems that we see in the city's water infrastructure in Jackson, Mississippi include lead pipes. So like many cities, Jackson is not unique in that it has lead pipes. I showed you the news article that Buffalo's got lead pipes. So it's not unique in that it has lead pipes, but it did not maintain the pumping infrastructure to maintain water pressure in those lead pipes. And so this allowed water to really stagnate in those lead pipes and accumulate much higher concentrations of lead and also microorganisms than in other cities that did have higher water pressure. It had gotten so bad in recent years that there were a number of periods of time where the pumps failed entirely, and so there was just no water pressure and no one could actually get any water. And when they fixed the pumps, it was still weak, and people had to run their water for long periods of time to wait for things to flush accordingly. And even then, it wasn't enough to really ensure good health. In 22 was sort of the last big crisis that Jackson faced with its water supply. So in late July, there was a big rainstorm that caused a lot of flooding, shut down the pumps. There was a boil water advisory. August 27th, another storm, more pumps failing, absolutely no water this time. And it took, so from July 29th until September 10th was extremely limited water supply for an entire city. An entire city that has not only all the people that normally live there, but also the flagship university for the state of Mississippi is also located in Jackson. So, imagine trying to do your chemistry labs with no water because there's no water pressure. How well would that go? Not great. Now imagine you're paying whatever it is the tuition of the University of Mississippi. Like, that's a lot of money. People were pissed. So funding was, emergency funding was eventually released. I think they used FEMA funds to get this back up and running. And there's been some new implements in place to make sure that these problems get fixed. I do believe that a portion of the bipartisan infrastructure bill specifically went to Jackson for the redevelopment of their water infrastructure. Okay, let's see. But fresh water, of course, is not the only water. Oceans, of course, are really important also. And one of the issues that we see in oceans is that materials can be transported around by the currents that exist in the ocean. So the ocean isn't just this sloshing surface that is on 75% of our planet. It actually has some direction and purpose to it. So one of the ways that we have been able to study and learn about the various ocean currents that we have is through accidental cargo spills. So there have been a number of accidental cargo spills that have helped to demonstrate just how moving and yet connected all of our world's oceans are. So a number of cargo spills have happened with Legos, and most famously with rubber ducks. So in 1992, so we start here in 1992. In 1992, there was a shipping truck coming from China going to the West Coast. And it was carrying a giant shipping container full of rubber ducks. This is a true story, by the way. I'm not just making up rubber ducks to be like juvenile, like I'm actually carrying a giant tanker truck full of rubber ducks. And there was a storm and it lost the tanker, or it lost the container full of rubber ducks. And it busted open as it fell over the side and was tossed about in the storm. And all of these rubber ducks came out of the tanker container. And over the years, people have been finding the rubber ducks from that tanker truck, or from that shipping container. So this was in 1992. Various landfalls have been made on the southern part of Alaska here, several years, as late as 2003, I think that says. They've even made it all the way over to Europe, at the East Coast. They've made it all throughout the Pacific Ocean. They're in various places. And so by following the patterns and the timing of these movements of rubber ducks, and because these were all standardized rubber ducks, we know how dense they are and how fast they should move in any given current of water. We've been able to sort of really map out these various ocean currents. We can use ocean currents to map things like how tsunami debris should be distributed. So this is a map of some debris from the 2011 tsunami in Japan. And as late as 2016, so 5 years later, we were finding pieces of debris from the tsunami here on the West Coast, and they were sort of following those ocean currents as we see them here. In oceans specifically, there are all of the same issues that we're worried about in freshwater can affect oceans as well. They can accumulate algal blooms, they can accumulate toxic chemicals, oil, sediment, garbage, etc. Microplastics, which is a new lecture topic that we'll be talking about, is a really big concern. And just to emphasize that point, this is a deceased bird whose stomach is filled with plastic trash. So this was a bird that was looking for things to eat, and it was seeing plastic things and mistaking them for food. And eventually its stomach became filled with plastic because plastic does not digest, and the bird died. We can see that plastic there. This map up here, this is looking at the consequences of that increase in biological oxygen demand that we saw in the Gulf of Mexico following the Deepwater Horizon oil spill. So this is the southern coast of Louisiana here. So this is the Gulf Coast. And there are, I forget exactly where the Deepwater oil derrick was, but it was out here not too far off the coast of Louisiana. But all of the oil tended to be concentrated right around here. And there was enough oil that there was an increase in microbial activity such that the biological oxygen demand skyrocketed and the dissolved oxygen available in the oceans dropped, especially at the bottom of the ocean. So this is a heat map showing oxygen concentration on the sea floor. So this is on the sea floor. and the seafloor and, some distance up from the seafloor. So if you think about how water bodies are shaped, right? So here you have, here's the top of the water, and then you've got the seafloor down here. So if you need oxygen to get to bottom, those oxygen molecules have to be dissolved first into the surface water, and then they have to make it past all the fish that are going to want that, and all of the other little microorganisms in the surface that are going to want it, and they have to get all the way down here. So in order for oxygen to get to the bottom of the ocean, it has to first be dissolved into the surface water, and then it has to not be used by anything else in the whole depth of the ocean on its way to the bottom. And so there's not very much oxygen generally left by the time you get to the bottom of the ocean. And now we've got this oil floating on top. And not only do we have that oil floating on top, but some parts of it are heavier than others, and it does start to slowly sink. So we have this oil kind of permeating the whole depth of the ocean. And so by the time you get down to the bottom, you end up with this dark red color, where here at the bottom, there is less than 2 milligrams per liter of oxygen available. In some places, especially immediately after the spill, they even measured as low as 0 milligrams of dissolved oxygen per liter of ocean water. And to this day, it is still comparatively hypoxic, reduced in oxygen concentration, still breaking down oil from the oil spill. Because not only do we have the oil from the Deepwater Horizon, but there are always just a myriad of little spills, never enough to really make the news, but just, in the daily process and goings about of oil derricks doing their job, some oil spills constantly. There's always some oil spilling constantly, kind of refeeding the whole cycle. And last but not least, one other issue that not only it also affects freshwater, but it specifically harmfully affects ocean acidification. When we think about ocean acidification, we think about it slightly differently than lake acidification. Not usually due to acid rain components, it's actually due to carbon dioxide. So carbon dioxide gets into water the same way that oxygen gets into water. It gets absorbed by the water or dissolved into the water, and then it percolates on around through. One of the consequences of an increased amount of carbon dioxide making it into water, because if there's more carbon dioxide in the atmosphere, then that's more to be dissolved into the water, right? We're sort of increasing the gradient of high to low concentration. So diffusion follows that concentration gradient, high to low concentration. So the more carbon dioxide there is in the atmosphere, the more carbon dioxide it is that will be dissolved into the ocean. As we increase the concentration of carbon dioxide in ocean water, one of the consequences of that is that it prevents organisms that need calcium carbonate from being able to form that calcium carbonate. So when you think about organisms that need calcium carbonate, you want to think about clams, mussels, crabs, lobsters, shrimp, any of those tasty sea organisms that have really hard shells. They use that calcium carbonate to form those really hard shells. And when you have excess carbon dioxide in the water, it prevents the calcium from it prevents those organisms from being able to combine calcium with carbonate to make that calcium carbonate. And so the consequence of that is that you get organisms with deformed shells, weak shells. They don't survive as long, they don't breed as well because they've got these weak shells. One of the proposed ways to help mitigate this, one of the proposed ways to help mitigate this is through aquaculture of some kelp, all different kinds of kelp. Because kelp, these are photosynthetic organisms. What do photosynthetic organisms make sugar out of? Carbon dioxide, especially carbon dioxide that's already in the water. They don't have to wait for it to diffuse in through the water. There's plenty of it all the way around them. So these kelps are proposed, and there are lots of farms up and down the northern East Coast, like Maine, New Hampshire, Massachusetts-ish, Connecticut-ish. They're doing this. They're growing kelp alongside mussels, clams, oysters, and things like that. And so the kelp are doing the photosynthesis, sucking up the excess carbon dioxide that's in the ocean water. This helps to raise the pH, which helps all the oysters and clams and mussels to recruit calcium with carbonate that they need to have more robust shells. So we're able to increase the pH of the water. We're able to get these organisms to make the shells that they need, and everyone is living a little bit more easily. And so there are all sorts of plans to try and do this and try and scale it up. It keeps being sort of a matter of funding and a matter of scale, because this does take kelp farming alongside mussels does take a lot of work and a lot of money. So it's getting there, but not quite. And then when I was putting this slide together, I did find this hilarious meme just to emphasize the problems that sometimes come along with, trying to, you want more photosynthesis? Well, this is the way you get more photosynthesis, but not quite doing work. All right, so it's a good meme. All right, so that wraps up this. And I realized that these transport lectures have been a little bit rambly in places, and so I added this slide to the slide deck. I did re-upload everything to Blackboard, so you have this. But as we start to think about our first exam coming up, I wanted to try and rethink about a better way to organize this. I'm usually tinkering with lectures a little bit, and I kind of realized how I should have tinkered with these two lectures a little bit too late. But you guys do have this slide, so you've got it on Blackboard. But if you wanted a different way to rethink about this to kind of direct your studying and direct your summary of information, I tried to collect kind of a lot of the overarching concepts on one side, and then we want to put our different sectors of the environment and then all of the little story times related to them in between there. So rather than interspersing overarching mechanisms with random story time, I wanted to try and put all the sort of actual pieces of information all together and then put all the story time over on one side. So hopefully that helps give some direction to how you might want to think about structuring your study for this. Does that help? Make sense? Okay. All right, so we've got a little bit of time that we can move to the next set of slides, try our best to get caught up on things. We're going to think about, so we've thought now about how toxins are introduced and move around in large sections of the environment, but now we're going to turn our attention to how do these toxins get in and move around an individual organism? How does an individual organism react to various chemical contaminants? And so that's where we're going to go next. I'll give folks a minute to transition if needed. I'll take a drink of water. All right. Does anyone need more time to transition? All right. So when we think about an organism and how an organism is going to deal with toxin introduction, we want to think about 5 different kind of points of 5 different junctures at which the organism's body will be doing something with that toxin. And so we call them the 5 sites of interaction, but they're not exactly specific locations. They're more like sites of point in time when stuff could be happening. And so we've got here our general model for these 5 sites. And so you can think of this box with all these arrows as an organism. This box with all these arrows represents an organism. And so when we think about how an organism is going to interact and deal with toxins, we want to first think about uptake. So toxins enter. How are those toxins getting in? Are they being inhaled? Are they being ingested? Is it diffusing through skin or other mucous membrane? How is it getting in? Once it's in, we want to think about that we want to think about, sorry, before I say too much about this action. So once it's in the organism, there are 3 different ways in which that organism can deal with those things. These 3 components here in the middle of the organism are not sequential. I've presented them as sequential because I have to, because time works in a linear fashion, I have to talk about something first and then second and then third. But within any organism, there is no action must happen first, metabolism must happen second, storage must happen third. We can go straight to metabolism, we can go straight to storage, we can go straight to action, right? We can do these things in any order. But I have to talk about them in some specific, in an order, because that's how time works. Okay, so we've ingested whatever the substance is, it's gotten in. And so now we have the option of, we can think about the action, how that particular chemical is going to affect the organism. What is the target within the organism that toxic substance is going to impact? Is it going to turn on some gene expression? Is it going to interfere with some cell signaling? What's it going to do? What's the action of that toxin in the body? And then related to that, how would we observe that this organism is being acted upon by some toxic substance? What is the manifestation of that toxicity? If we're not going to do action, we can think about how this organism is going to metabolize the toxin. The metabolism is the organism acting upon the chemical, dealing with that chemical. So action is the chemical working on the organism. Metabolism is the organism working on the chemical. How is this organism going to digest it? How is it going to break it down? What are the consequences of the ways? What are the consequences of ways in which an organism might break these compounds down? We're usually going to be thinking about this with an eye towards the organism's metabolism is probably trying to detoxify whatever that substance is. How successful are they at it? What are the consequences of being not successful, etc. Third option for once it gets into an organism is we can think about the storage of that compound. When we think about storage of a compound, we're usually thinking about that compound is still there, but it's inactive, it's sequestered. It's there, the toxic substance is in the organism, but it's not having an action, and we're not able to metabolize it. We've just sort of pocketed away somewhere. And then last but not least, the organism will finally excrete either the direct toxic compound or the metabolized, broken down product of that compound. So once again, you have to ingest the organism and then ingest the toxin into the organism. Once we're in the organism, these 3 sites of interaction here, they're not sequential. And so you'll notice that those arrows are really going all over the place. There's no one way to go on this path. But the end is always excretion. All right, so this is our general model, and then we'll see some of the details as we go through the general model. All right, folks good? Okay. All right, so our movement through this model is going to come back to that KOW concept. Generally speaking, when we have a low KOW, that suggests things are lipophobic, i.e. hydrophilic, and they will flow through quickly. The higher the KOW, the more lipophilic, i.e. hydrophobic, the more slowly it's going to move through those 5 things. And oftentimes with lipophilic substances, excretion depends on some metabolic transformation, converting it to a water-soluble state. Right, because we're usually trying to get it out. Most organisms have some mechanism to urinate. Not every organism necessarily has a means of defecation. We're fortunate in that we have both and they are separate. Some organisms try to combine defecation into their urination. So it's not always a clear-cut way. We also want to think about the toxicokinetics which include uptake, distribution, and metabolism of that substance throughout the organism's body, i.e., how much is going where. Right? Because all three of those states can also be simultaneously happening. Some portions of the, kernel of toxic substance that have been ingested, some could get stored, some could get metabolized, some could be exerting an action. Is the substance more likely to be stored? Is it more likely to be metabolized? Right? We're going to be doing all the things all at once, and the question is going to be kind of where does the majority go? And last but not least, we also want to think about the toxicodynamics, which is especially relevant to the action site. And so we're going to be thinking about the kind of reaction that we get and how intense is that reaction, the degree of reaction. Is it a immediate and severe reaction, or is it just, kind of a little tummy ache for a mouse? Does tummy ache happen instantaneously? So all of this is really going to be dependent on not only the compound itself, but also what kind of organism it is, where it lives, what its physiology is. There's going to be no one way to really think about it. All right, everybody good? Okay. All right, so let's talk about uptake. For most organisms, most exposure of chemicals to most organisms is going to happen through passive diffusion. You're just sort of going through the environment, and you are moving through a concentration gradient where there's a high concentration of some toxic substance outside you, there's a low concentration of some toxic substance inside you, and the toxic substance will move down that concentration gradient from high to low. This is especially true if you're an aqueous organism or some other cellular-based organism, like you exist as unicellular or a colony of cells, where you get things through mainly phagocytosis, where you're sort of bringing in bulk things. It depends on how soluble that compound is. It's lipophilic, lipophobic. If there's any sort of form that is going to be preferentially absorbed. So, for example, 2,4-D is a common insecticide, but 2,4-D, whether it's in an aqueous or non-aqueous environment, exists in slightly different chemical forms. And so in some organisms, in some habitats, one form is preferred. In some organisms, in other habitats, the other form is preferred. So which is it that's happening? Weak acids tend to be preferred. Weak acids like this tend to be preferred absorption in slightly acidic environments. So if you're in a water body that is experiencing some acidification, either from acid rain or excess carbon dioxide, then that could make the uptake of this form of 2,4-D more easy than this one. All right. Everybody okay here so far? Okay. All right, so we're in. Now we're going to think about how this substance is likely to be distributed. So vertebrates tend to distribute compounds through either the digestive system, right, going through the alimentary canal, the inner tube to our body's outer tube, or through the respiration system. In either case, whether you're going through the digestive system or the respiration system, usually the final destination for things in both of those pathways is the bloodstream. So most things are always getting to the bloodstream. How easy it is for a substance to go from the digestive system to blood or the respiratory system to blood are going to depend on, of course, KOW. We might expect more. We might expect more lipophobic kind of substances to go through the respiratory system, right? You're breathing things in. It's got to cross the water moisture barrier that's in your alveoli in order to go into your bloodstream. If we're in the digestive system, there's a lot of lipids going on there. We might expect more hydrophobic substances to be more easily introduced into the bloodstream through the digestive system. So again, think about what kind of compound it is. Some compounds have the ability to cross the blood-brain barrier even. So once it's in your blood, it can work its way up to your brain if it's of the right chemical constitution. Similarly, in invertebrates, we can think about some of these same concepts from vertebrates and invertebrates. But remember, blood in invertebrates is usually that hemolymph stuff. And especially in insects, we don't exactly have lungs or alveoli, but we usually have the spiracles. Other kinds of invertebrates will have other respiratory systems. And of course, we can't forget plants. Plants also get exposed to toxins, and we need to think about how stuff's going to move around in plants. So when we think about plants, we're going to be thinking about especially the apoplastic root, which is the space outside of those membranes. So apoplastic, these are sort of the watery interstitial spaces that go around various plant cells. So as roots, I can draw this, but not on the screen. So for those watching at home, you won't get this drawing. But the apoplastic root so all right, so imagine we've got a root, the root of a plant. And that root of the plant is in the soil. And there is water and all sorts of stuff in the soil trying to get into the plant. Everybody with me so far? Can everybody see outside? Okay. All right. So as water, right, there's more water outside the plant than there inside the plant. Everybody remember from Bio 110 when we talked about the water potential of moving substances, especially water through a plant. There's more water outside than inside. So we have high concentration outside, we have low concentration inside, and so the water is going to move into the root. And as it goes through the root, it's going to go past all of those cortex cells. I wish I had more chalk colors. All right, so there's all these cortex cells in our roots and our water and other substances. They're going to work their way apoplastically outside of those cortex cells. They will not be entering the cortex cells. Why aren't they entering the cortex cells? Because it's difficult for substances to cross a plasma membrane, whether you're hydrophilic or hydrophobic. It's hard to cross a plasma membrane unless you're really small, that you can slip in between those phospholipids and sneak past everybody's hydrophobicness. It's really hard to go into the plasma membranes of things, especially if you're a plant cell that not only has a plasma membrane, but also a cell wall, right? That's just a whole pain in the ass to go through. And water and anything dissolved in water likes to take the path of least resistance. And so they're going to stay apoplastic and go around all of those plant cells until, of course, they hit Casparian strip, that boundary that hopefully will stop the toxic substances. Sometimes the toxic substances get through the Casparian strip, sometimes they don't. So when we think about xylem, because that's our main way into the xylem is through the Casparian strip in the roots, So it's a lot easier to use apoplastic, and especially if you manage to even get into the xylem, those cells are dead. There is no more plasma membrane through which to pass. It's just a giant straw of dead cell wall. All right, we've got our xylem. Things tend to be watery and move through there. Phloem, those cells are living. And so if you want to, if a chemical needs to move through the phloem, it does need to move symplastically. It needs to move within the membrane, within that cytoplasm of adjoining phloem cells. Those cells are living. They direct where the phloem is going. It's also more sugary, right? Sugar is, on our spectrum of totally hydrophobic to totally hydrophilic, sugar is a lot more hydrophobic. It's not totally hydrophobic, but it's comparatively hydrophobic compared to water interacting with water, right? So it's on that spectrum. So we tend to want to think about more lipid-loving compounds interacting with phloem and more water-loving compounds interacting with xylem. All right. All right, so we've gotten in, we've ingested, and we've distributed throughout our organism's body. All right, so we're going to take storage first, because storage is pretty straightforward. So we tend to think about storage not with every compound, but with more of the lipophilic, lipid-loving compounds. With vertebrates, we tend to see toxins get stored in lipid tissues, and then once those lipid tissues are sort of called upon to deliver some energy, that's when the toxins stored in them are released. That can happen due to environmental stressors, breeding pressures, all sorts of things. So one of our sort of case examples of toxin storage in organisms and how that can affect things comes to us from puffins. So this is a puffin. They are cute. These are seabirds. They live in very northern climates on the ocean. They like cliffs, rocky places where it's really hard to get to. They're kind of like northern hemisphere penguins a little bit, right? Like penguins are southern hemisphere, puffins are northern hemisphere. All right, so puffins, like their penguin counterparts, love fish. Really oily fish. And these fish can bioaccumulate lipophilic toxins. So one characteristic that is fairly similar across cold water or cold habitat organisms is they tend to be a much more fat-based food web, right? They're using fats as storage. They're using fats as their primary food source. Higher the fat content, the more good a food source it is, right? If it's cold out, you want all the lipids you can get. So these little fish are very oily, lipid-based. They're sort of sardine kind of fish, very oily, and they will bioaccumulate lipophilic compounds like dieldrin. The dieldrin is an insecticide. It's often sprayed on crops, but it's also sprayed for pest control, especially mosquitoes. Even in cold places, especially in the summer, you can get really serious mosquito problems. So dieldrin is an insecticide that is sprayed a lot, and it can wash into water and accumulate in these fish, which then the puffin gets and eats quite a lot of. So puffins live solely on the oily little fish, and so they will bioaccumulate that dieldrin from these little fish. And what we have observed in puffins is that when times are good, when there's plenty of fish, nice and warm, habitat safe, no storms, when everything's good, the concentration of dieldrin in the blood is actually very low. And so when we think about blood concentration, this is the amount that's like sort of circulating and moving and has the most potential for interaction. But if you take a lipid biopsy, a fat biopsy, you would see that the concentration of dieldrin in fat deposits in that puffin would be very, very high. All right, so that's good times. And then in bad times, there's not a lot of fish. You're trying to mate, you're trying to sit on an egg and you can't go hunt for fish. It's extra cold. Maybe there's a storm. Things are not great. When it's lean times, right? When it's lean times, those puffins are going to call on their fat deposits. They're going to start to mobilize those, break down those fat molecules into sugar to do cellular respiration. And when that happens, we release the dieldrin that was stored in the fat into the blood. So this stored fat dieldrin in good times becomes the circulating blood dieldrin in lean times. We not only see this in puffins, but we see this in other birds that experience something very similar, especially if those birds are migrating over long distances, like these ducks. Ducks travel very large distances, and they are much more fatty than like a chicken, because they've got to have those energy storage to get them from point A to way the fuck over there, point B. All right, so that's storage. Oh, all right. And in 10 minutes, we're going to do metabolism. All right, we can do this. All right, so I'm just going to throw everything up here just to make sure that I don't miss anything. And that's actually it. Okay, so metabolism. So organism ingests something, you've uptaken it, you've distributed it. Option B, if you're not going to store it, is to metabolize it. The metabolism of toxic compounds generally takes 2 phases. Phase one is usually some initial enzymatic transformation. So an enzyme will come along and take whatever compound we're trying to break down, and we'll try to oxidize it, we'll try to hydrolyze it, break it into smaller pieces. And the ultimate goal of either oxidizing or trying to hydrolyze and break apart into smaller pieces, the ultimate goal of doing so is to add hydroxyl group. So this, remember, this is our hydroxyl group. Remember I said know your functional groups? So we want to try and add a hydroxyl group. We want to try to increase the lipophobicness. We're trying to add polar pieces. The more polar we can make it, the more points of attack your later metabolism has on that compound. If it's a totally lipophilic compound and there's no way for your watery enzymes to kind of get in there and break things apart, then it's a really hard compound to metabolize. But if you can do a little enzymatic transformation and just get a notch in that armor and throw a hydroxyl group on there, that makes it easier for later metabolic actions to try and break that down. So that's what we're trying to do in phase one. There are some potential consequences of doing this, of breaking apart compounds, adding hydroxyl groups. So just doing this isn't totally neutralizing the compound. This is just sort of step one in attempting to neutralize the compound. For really toxic substances, what can sometimes happen is you can, through this oxidation process, turn that compound into an acetylcholinesterase inhibitor, which we've seen the problems that causes. Or we can sometimes end up binding it to our DNA. We can bind it to our DNA, and it can upregulate, downregulate, inhibit, promote, right? Depends where on the DNA it binds, what gene it's bound to, what's coming before the gene, what's coming after the gene. Maybe you get lucky and you bind it to a histone and it doesn't do anything. So for particularly toxic compounds, there are some consequences to trying to do this. All right, but if you make it through phase one okay, and you slap on this hydroxide group, and you're ready to go to phase two. Phase two is just this general further enzymatic activity. There's a whole bunch of other options that we have in phase two, which we'll talk about a little bit later. The goal of phase two is to further make this a hydrophilic compound, a lipophobic compound, right? We want it as watery and dissolvable as possible. When compounds make it to and through phase 2, if we suspect that, let's say we're testing organisms for potential contamination or exposure to some toxic compound, if those organisms have already been exposed to that compound for kind of a long time, they might already be well into phase 2 of their metabolic breakdown of that compound, meaning that there might not be terribly much of it left. There might only be these sort of broken down residual bits of that toxic compound. And so it becomes very difficult to chemically detect what the toxic compound originally was after it's been through phase 2 of our metabolism. Sorry, was everyone good here? Okay. One example of what can happen in phase 1 that we can see is with Aspergillus fungus. So Aspergillus fungus is a fungus that can contaminate corn and other grains, and it produces a toxic compound called aflatoxin. And aflatoxin comes in 2 flavors, B1 and B2, and particularly the B2 form is much more toxic to vertebrates than the B1 form. And it's specifically because of this center part here. One of the common ways that phase 1 introduces hydroxyl groups, is through a collection of genes known as the cytochrome P450 genes. So cytochrome P450 genes do a lot of that phase 1 metabolism of trying to put a notch in the armor of those lipophilic compounds to try and break them up and become more watery. So cytochrome P450 will create, those genes will get turned on. They'll make a bunch of enzymes like CYP3A. And CYP3A will take aflatoxin and cut out this middle bit. This middle bit then goes on to further metabolism. And depending on how your further metabolism deals with it, it could potentially turn it into this form, which is cool. You can kind of waste that out. Or it can turn it into this form and toxic, add some toxic activity to your liver. So depending on what form of aflatoxin, depending on how CYP3A deals with that middle part of the aflatoxin, you can get potential toxicities. So just trying to metabolize these things isn't always necessarily a guaranteed cure-all. All right, with that, we are out of time. We'll leave it there and we'll pick up with more metabolism stuff next time. I will stop the recording. There we go.